Welcome everyone, welcome to our live audience and welcome to our YouTube audience as well. Today we have the pleasure of welcoming Dr. Kent Lloyd to our webinar series. Today Dr. Lloyd will be going over the Mutant Mouse Resource and Research Centers, MMRRC National Consortium. Dr. Lloyd is a veterinarian and research scientist with expertise in embryo manipulation, genome-wide mutagenesis, and comprehensive phenotyping of the laboratory mouse. His primary research focuses on development of mouse models of human disease, genome editing, and preservation and resuscitation of genetically altered mice. Currently, Dr. Lloyd serves as the director of the UC Davis Mouse Biology Program, and he is also the PI of the MMRRC at UC Davis, which we'll be learning more about today. Without further ado, Dr. Lloyd, I'd love to hand the microphone over to you so you could get started. Thank you again for joining us today.
All right, Devin, thank you very much, and thank you to the organizers for the opportunity to be able to tell everyone about the Mutant Mouse Resource and Research Center Consortium, the MMRRC. We were established now almost 20 years ago by the National Institutes of Health, specifically currently through the Office of Research Infrastructure Programs. And just a plug there for ORIP, or the Office of Research Infrastructure Programs. As I mentioned, this is the funding agency within the Office of the Director at NIH, which provides support for the MMRRC Mouse Repository Consortium. So to just give you some general background, the MMRRC, as I mentioned, it's a consortium, and there's 4 regionally distributed archive and distribution centers within the consortium, and also an informatics coordination and service center, the ICSC, which functions as a fully integrated repository system. I know all of you have heard about the National Primate Research Centers, Resource Centers. This is the mouse equivalent to that national system. So what our centers do basically is to import, conduct quality control, maintain, archive, and then distribute mouse lines upon request to the biomedical research community. The ICSC provides an online searchable catalog, a dynamic website, strain curation services, data resources, technical assistance, and outreach and education activities. To give you a little history of the MMRRC to put it all in context so you can understand why this is such an important repository system and the role it plays in our research community. Back almost 20 years ago, it was recognized that the number of genetically modified mouse strains was expanding greatly, and it was resulting in increased pressure to preserve and make these mice available for research if you consider the amount of investment that was being put into them for why it came to be. And I explained in 1998 where the discussion really started to happen in terms of the exploding number of mutant mouse lines that were being produced in individual laboratories. And the NIH in particular wondering how are we going to preserve, protect, and maintain these lines in perpetuity for other researchers to have access to them, because they were being made as a national investment. So in March of '98, the NIH held a meeting on priority setting for mouse genomics and genetics resources, which really set the stage for the origin of the MMRRC. As a result of that meeting in 1999, the National Center for Research Resources, NCRR, which was one of the institutes at the NIH, it's now sunsetted and it's been replaced by ORIP, established the MMRRC to serve the scientific community, and they offered U42 cooperative research agreements to develop animal model resources. So it was an open call for institutions to apply in order to fill this need. In May then 2001, the MMRRC was launched and began accepting strains, or mice obviously. In October, 1 year later, a little bit more than a year later, the first strains were then being made available to requesting investigators. In 2003, we added embryonic stem cell lines to the collection, because as we found that there were even a greater number of genes that had been mutated, had not yet been converted into mice, but they could be readily converted into mice from embryonic stem cells. So the MMRRC, within 2 years of being launched, had already begun to evolve and go beyond just merely accepting live mouse strains and began to accept embryonic stem cells. And this was really launched with the importation of the very large Bay Genomics gene trap clone collection, which we still have and make available to researchers. And at that time in 2003, we also started offering cryopreserved embryos to be distributed to investigators upon request. That made the shipping process a little bit easier, especially if investigators had the capability of recovering embryos to live mice in their laboratories. In 2005, then the MMRRC was recognized internationally and became a founding member of the Federation of International Mouse Resources. And a lot's happened between 2005 and 2015 with the growth of the MMRRC, but something I want to point out that the MMRRC has had a big role to play in, and that is in the rigor and reproducibility challenges that we all know about in biomedical research. And we, as a consortium, published this commentary piece in Nature on the use of mouse biobanks and basically warning everyone we need to use these repository mouse biobank systems, or we may end up losing them to our own peril. So that's the history of the MMRRC. As I mentioned, it's a consortium, and this is a map that's showing the locations of the 4 regionally distributed resources. I'm at UC Davis, and so there's one here at UC Davis. There's another at the University of Missouri, Columbia, another at the University of North Carolina, Chapel Hill, and also an MMRRC located at the Jackson Laboratory. So it's separate from the Jackson Laboratory itself, but it's within the Jackson Laboratory institution. In addition, the ICSC, as I mentioned, is also currently at UC Davis. So what is it we do? Well, in a nutshell, we accept donations of mouse strains from individual researcher laboratories. We then maintain colonies and cryopreserve these strains as germplasm, being sperm or embryos, and or as embryonic stem cells. We distribute live mice, either as that are being maintained in active colonies or cryo-recovered from frozen libraries. We also distribute frozen germplasm and ESLs upon request. We are a supplier to both academic researchers and to for-profit entities. I can get into that in a little bit. Our NIH funding supports the importation, maintenance, and archiving of the lines, but then we charge investigators a nominal fee who request lines or ESLs or germplasm for distribution. So it's as I mentioned, it's a cooperative agreement. We get NIH funding in order to get the mouse lines in the door, quality controlled and maintained, but then we charge investigators a small fee when they go to request those out of the repository. We also, though, provide resource support services such as microbiome analysis, SNP genotyping, microinjection electroporation, and other services that are provided by the host institution. So again, 4 MMRRCs located at 4 host institutions, and they open their doors to investigators who want to have access to these other type of services. So there's really a leveraging then of what the institutions have in addition to their MMRRC to provide help to investigators who request it. And then the MMRRCs are also involved in developing new technologies to improve utilization of mutant mice, such as improving artificial reproductive technologies, cryobiology, genetic analysis, phenotyping, and infectious disease diagnostics. This graph just goes to show since 2011 the growth in users of the MMRRC. Now, this includes both donors as well as requesting investigators, and this is cumulative over time, as I mentioned, since 2011. So number of users on the y-axis, year on the x-axis, and you should see the total users here in blue are upwards of almost 11,000 investigators who have used the MMRRC, and the orange shows the growth in the number of new users on an annual basis. This graph shows distribution activity based on the format of the mutations that are distributed from the MMRCs over time. So the number of orders are shown on the y-axis and year along the x-axis. And the colors and lines are, as shown in the legend, the different formats of mutants that are distributed. So in blue are the cell lines. You'll notice the cell lines, such as the ES cell lines, were very, very popular when they were first deposited back in 2003, but they're beginning to wane in popularity, as you see in 2007, 2008, 2009. Their distribution numbers have gone down, but they were very, very popular early on, while mice have continued to climb in popularity, and orders have continued to now rise and look like they're plateauing. The black number, the black line on the top, is just a sum of all the different formats that are distributed by the MMRRC, be they cells, embryos, sperm, mice, or colonies resuscitated from a cryopreserved archive. This next slide also shows distribution activity with regard to the academic institutions that are served. So number of academic institutions on the y-axis, year on the x-axis, showing a very healthy number of academic institutions that we serve annually, more than 500 on an annual basis. And more on distribution activity in this slide, in which we serve not only the community here in the United States, but really all over the world. And so we have mostly requesting investigators, but even donating investigators from 43 countries and territories from around the world, and that continues to grow. Very importantly, though, science is obviously an important part of this, and so we want to contribute to science, and we monitor that by looking at how many of our MMRCs are included as either co-authors or, in most cases, cited as by authors who cite the MMRC as the source of the mouse lines used and published in their research papers. So x-axis, number of publications, and the year is shown on the x-axis, and showing a good, healthy number of publications on a regular basis, which is steadily increasing. We're not finished with 2017 yet, so we expect this to be up in the range of where we have been over the last couple of years. So this is obviously a very, very important metric that we follow, because we aim to be contributing to new knowledge and contributing to the advancement of science. So what are the benefits to you as a depositing investigator? That is someone who has made a mouse line in your laboratory and decides to donate it to the MMRC and deposit it there for archiving and distribution. I want to point out and remind everyone that you retain ownership of this line. We merely act as stewards of your line, and you give us permission then to distribute it. You can give us permission to distribute to both academic and for-profit entities, or you can restrict distribution to only academic entities. That's up to you. So the benefits to you is that using the MMRC helps you to fulfill your NIH obligation, if you're funded by NIH, to share biomedical research resources. In fact, that is a requirement for in many RFAs and RFPs and grants that you have an indication of how you will participate in resource sharing. And so including a statement into your grant, and we can help you with that, to indicate that any mice that you create as a result of your grant will be deposited in the MMRC, and that helps to fulfill your sharing obligation with regard to resources. And again, as I mentioned, the MMRCs are available to help you write that part of your grant. It also benefits you by reducing your need to maintain mice and utilize what might be limiting animal housing at your institution. Because we are a repository system, we work together in the 4 host institutions, and we have really an unlimited supply of housing capacity to be able to maintain mice. So this takes the burden off of you and allows us to assume that burden. It also then, of course, then reduces your per diem cost liability by getting those mice off of the shelf at your institution and putting them on ours. We create a cryopreserved archive, and we do that to ensure the safety, the protection, and the perpetuity of your line, not only for others to use, but also for you, in case you say, you know, you may be done with this line for now, but you may want to use it in the future. Well, it's there and available for you to access at any time in the future. It also relieves your burden of shipping mice to investigators or collaborators that contact you. I know many of you who have created mutant lines and published on them, and you get requests from friends, collaborators, and investigators you may not know from around the world who would like to have your mouse line. And by depositing them in the MMRRC, we can then take on that responsibility for you, so you don't have to worry about any of that. And then also, as one other benefit, is that you can receive a one-time free delivery of either live mice, if they're currently on the shelf, or germplasm for every line that you donate. So that's kind of an incentive to encourage you to donate your line. And you only need to pay the shipping charges for whatever the format is that we ship to you. There's 3 ways to deposit mice, and most of you will be depositing through our Type 1 category. And this I call NIH funded, and this is where you as the donor fill out an application. That application then undergoes scientific and administrative review, and the acceptance is determined by a coordinating committee that meets monthly. The coordinating committee is made up of the PIs of the MMRRCs and the ICSC, of NIH staff, and occasionally scientific consultants that we reach out to, including our external advisory board, to give us feedback on particular mouse strains. There's also a Type 2 mechanism, and again, it's also NIH funded, and that is a mechanism by which an NIH program officer can nominate lines to be accepted by the MMRRC, and those go through a direct acceptance process. So why this is good is that we then help to encourage different institutes at NIH to take it upon themselves to either nominate lines either by internal NIH investigators or their external investigators that they fund, to encourage them to get lines into the MMRRC. So what the MMRRC does, it provides the institutes a certain number of spaces or slots every year that they can fill, and all they need to do is to nominate, you know, Professor X's line to be deposited in the MMRRC, and then we take that right in without review. And then there's the Type 3, and I call this investigator-funded donation. And that's where an investigator funds fully the importation, archiving, and maintenance costs of the line. Now, you might ask, why in the world would anyone do that? Well, sometimes investigators have large collections of lines that they'd like to deposit, and instead of going through one by one the acceptance, where some might be accepted, some not, based on the review criteria, this overcomes all that, and you can provide the funding to directly deposit in the MMRRC system. It also allows some lines that might not meet all the acceptance criteria to be accepted, and that way we don't use NIH funds to import, archive, and maintain, but we use the depositing investigators' funds. And also, it's a means by which we allow investigators who might want to use the MMRRC infrastructure in order to hold lines for them strictly, so that they can maintain them, cryopreserved, and protect them, and have it for their own use. And so the MMRRCs allow that as well. Again, we don't use NIH funding for that. That all comes from investigators funding themselves. So you can choose which of the mechanisms on your application form to use. Now, with regard to review criteria, this is very important. As I mentioned, the mouse strains are reviewed monthly by the MMRRC Coordinating Committee. The reviewers use the information that is provided in the strain submission application, and they assess a set of objective criteria necessary for acceptance. And those criteria are listed here. The 6 are listed here. First, we go through and evaluate whether the line was created using sound scientific methods. Secondly, we determine whether the strain is distinguishable genotypically and or phenotypically from other known existing mouse strains. Thirdly, it's important that the line exhibits a clearly identifiable and or useful, research useful phenotype. Number 4, it's also important that the line exists at no other distribution center. So once a line is accepted in MMRRC, it's only distributed from there. A line from the MMRRC cannot also be distributed from another center. Fifth is that the line is available for distribution to the scientific research community, and the implication is that when you deposit, again, you have a choice to academic only or academic and for-profit, and that you agree that we will conduct all the distribution, and you turn over any requests for your lines to the MMRRC to take on that distribution activity. That's very, very important. We wouldn't want you to be distributing the mice at the same time the MMRRC is trying to do so as well. And then finally, it's also important that controls are readily available, or unique controls will be provided with the submission. So as long as the lines fulfill those 6 criteria, they are generally considered acceptable for admission. And applicants are usually notified by mail within a week of decision, which is usually within a few weeks of an application. So there's not a whole lot of wait time on the part of the applicant. Okay. All right, well, that's the donation side. What about the requesting side? What are the MMRRC benefits to you for this? Well, you have access to unique models that are otherwise not available commercially, or even from other potential repository systems, because again, the strains that we distribute are unique. We only distribute specific pathogen-free health status mice, and several of our repositories are approved vendors, and I'll get into that in a moment. We also provide the highest level of genetic quality control and access to special resources, support services, and also technical support throughout the process, both in obtaining mice, donating mice, and once you even obtain the mouse, if you have further information that we can help you with on using that mouse, we provide that as well through the ICSV. I mentioned approved vendor, and what this means is that an institution has recognized the MMRRC as maintaining mice at a high enough health quality that mice from the MMRRC can be directly imported into a vivarium at your institution, and foregoing quarantine or other restrictions imposed on that during importation. So that's very important. And so it says a couple things. One, it means that the MMRRCs that are recognized as approved vendors are operating at a highest level. And secondly, what's important is that it helps you to know that when you obtain a line from us from the MMRRC, and if your institution has recognized us as an approved vendor, then you have access to your line far faster than if it had to go through quarantine. If you remember earlier, I mentioned too that not only do we accept lines from individual investigator laboratories, but also we accept what I'm calling major collections. That would be large group of lines, like, for example, the Bay Genomics collection was a large group of ESLs that we accepted in one tranche. We have many, many major collections that have been deposited in the MMRRC. And as you can see here, a list of, I think most of them here, the UCOM collection from BASH, Bay Genomics Beta Cell Biology Consortium, and our most recent one is the COMP collection from the COMP IMPC project. Again, all of these are available to you and can be found on the catalog. And we'll get to that in just a moment. I want to also emphasize the fact that we have a we're very much committed to rigor and reproducibility, and we do that through a number of ways. With regard to a critical assessment of incoming strains, all submissions require a detailed information about the method of genetic manipulation, genotype and genotyping procedures, genetic background, such as the source of the strain, ESL used in development, number of background generations, and backcross parent strain, and also reproducible phenotype. We also look for a thorough strain description, which is, we hope is provided on a strain detail sheet, and then we make that available to you. And this strain detail sheet includes the strain name using established nomenclature guidelines, gene details, strain description, research applications, the origin, colony husbandry information, a whole host of very important information. The primary references for the strain are also available through the PMCID or PMID links, and the MMRRC maintains contact with the submitting investigator or the donating investigator to aid in troubleshooting phenotype reproducibility. And the phenotype information that arises in the literature from use of these strains is added on a regular basis. Now, in addition, all the strains are assigned an RRID, or Resource Identifier Number, which is a globally unique accession number allowing authors to cite the resource that they use in the material and methods section of their manuscript. So if a strain that is submitted to donated to us doesn't already have an RRID, then one is assigned from that point forward, and you can use that RRID in your manuscript when you publish on that, so that others know precisely what strain that you use for your research. The genetic identity of all strains is confirmed upon receipt of the donated mice and prior to shipment of animals or materials to a requesting investigator. The genotyping assays are optimized and standardized protocols available to ensure the investigators have a means to genotype any MMR strain that they receive and maintain. Provide additional genetic information for congenic strains, background strain information that could all be ascertained using a MegaMuga, which is a low-cost, high-density genotyping platform. This may be especially critical to investigations using incipient congenic strains, such as between N5 and N10 generation, that are not fully congenic. Many MMR strains, including the highest demand strains, already have this information provided. MMRRC strains are maintained with strict biosecurity SOPs and rigorous health monitoring programs with extensive exclusion lists that include all known pathogens of mice and the vast majority of opportunists. Health monitoring tests use state-of-the-art methodology, and the MMRRC can test for agents not on its exclusion list. There's also detailed descriptions of the MMRRC facilities and husbandry conditions, such as housing, diet, bedding. And then also, we try to ease the access and use and obtaining the mice by the use of an MMRRC conditions of use agreement. And that encourages the procurement of animals directly from the MMRRC in an effort to ensure rigorous genetic and biosecurity control of strains. It can also avoid issues of genetic contamination, misidentification strains, and changes in animal health that can occur in independent institutions. So we don't use, in most cases, we don't use an MTA, but this is an electronic conditions of use agreement, which simplifies the process. The MMRRC also provides numerous value-added services that can aid investigators in troubleshooting phenotype deviations and ensuring reproducibility. As I mentioned before, they include genotyping, karyotyping, speed congenics, health monitoring, microbiome analysis, and an array of phenotyping tests. And then also the MMRRC scientists and veterinarians at the host institutions for the MMRRCs are always available to consult about the use of MMRRC strains. I'm going to show you a couple slides just about UC Davis's MMRRC to give you just kind of a flavor for the kinds of things you have access to beyond just obtaining the strains. So here's a list of value-added services that you can use from UC Davis as a result of interaction through our MMRRC. That includes everything from rederivation, cryopreservation, cryorecovery, colony expansion via IVF, germline transmission testing, and so on. But it includes everything: molecular biology, phenotyping, and a sundry of a number of different tests and procedures. I want to point out a couple here that I think might be of interest to you. We've just rolled out what we call the Speed Cohort Delivery Service. We call it SCD. And this combines genome editing using Cas9 and artificial reproductive techniques to rapidly create mutants, expand colonies, and produce sex balance and age-matched cohorts appropriately sized for research. So as you can see here, if needed to, we could take an existing mutant strain and further modify it using CRISPR, or merely take the mutant strain that is present as germplasm and reestablish that by IVF, and then go on and produce experimental-ready cohorts from rapid IV expansion. And the deliverables here would be homozygous and wild-type mice that are age-maxed of both sexes and include extra heterozygous breeders. And as you can see, this can take place within a matter of about 33 weeks compared to 18 months using traditional expansion methods. Another service that we've rolled out is what we call fecal archiving and analysis, and that's using the gut microbiome to address rigor and reproducibility. And the way this works is we send what we call a fecal collection kit to the depositing PI. And so in this very small box, simple kit, there's instructions with pre-labeled vials, picks, return labels, and a reusable insulated box. The PI then collects the samples prior to shipping their mice to the MMRRC. This would be a donating investigator prior to shipping their mice to the MMRRC. This is a line that was accepted by the MMRRC. They would collect those samples and ship them to us. The MMRRC then receives, records, and archives the samples. And we offer microbiome analysis to any requesting or recipient PI. And then we ship the mice to the PI, and we can send the fecal collection kit to the recipient or requesting PI to compare how the microbiome differs from the original colony, from the donor, and the colony now established at the requesting investigator. And that helps to allow the requesting investigator using an MMRRC mouse to assess the role of the microbiome and phenotypic differences that they might observe between the depositor and recipient laboratories. I mentioned earlier too that one of our most recent collections is from the Knockout Mouse Production and Phenotyping Program. I'm sure all of you have heard about it. And I thought I would just point out one or two things that might be of particular interest to neuroscientists. As you know, the COMP2 is part of the International Mouse Phenotyping Consortium. And the project, just to give you, those of you who don't know, it's a global infrastructure project recognized by the G7. Our goal is to create 20,000 knockout mouse strains on a single genetic background strain, and that's a C57 Black 6N. We're characterizing each strain through a standardized and harmonized set of phenotyping protocols. We're integrating the data to an existing mouse and human disease data resources and providing those strains and phenotype data for use by the research community. And of course, that's where the MMRRC comes in, because those strains from the COMP project are all put into the MMRRC. The phenotyping data is available from the IMPC website. So here's an example for nervous system phenotypes. Around 5% of the tested genes with null mutations have a nervous system phenotype. And you can see over here on the right the different types of phenotypes that have been recognized in those lines. And about 27% of tested genes with null mutations have a behavioral or neurological related phenotype. And again, over on the right shows you a list of the kinds of phenotypes that have been observed during the IMPC phenotyping program. So I'm telling you this because these lines are the kinds of lines that then would be available in the MMRRC to you. So here's one example. This is the ferrochelate reductase 1-like gene. It encodes an outer core component of the AMPA receptor in the brain. It's a protein thought to interact with inner core components of the receptor and plays a role in modulation of glutamate signaling. And mutations in this in humans have been associated with early infantile epileptic encephalopathy. And in the COMP2 IMPC program, it was found that this gene had exhibited extensive pleiotropy and also sexual dimorphism. And I'm just showing here a few of the phenotyping results that we're seeing. First of all, there was a reduced survival with only 10% of homozygous surviving to adulthood, a reduced grip strength, and hyperactivity in the open field. But this is only just scratching the surface of the different phenotypes that are available. Again, I point these out as an example of one of the major collections held by the MMRRC as mutant mice that you have now direct access to. So I think it's a very useful resource. Well, as we get to the close, let me go through some practical aspects of how do you go about getting mice from the MMRRC. Well, this is a screenshot of the homepage at www.mmrrc.org. If you go to the homepage, you see this dashboard, and in the middle is a search box where you can search our catalog by any means that you like, either using major mesh terms, the exact MMRRC ID if you happen to know it, a gene or a strain name, the MGI ID. You can search by major collection, or you can search by the RRID. And then once you do that, the catalog appears here. You'll see that on the left column, this is where you can add a gene to your cart, a mouse line to your cart. Next column is a description of the strain and cell or cell line name, which you can click on to access the strain details sheet, which has all the information on the strain. This is the ID number, the RRID number for the strain, the availability of that strain and any restrictions. So for example, this first one is in the cryoarchive. The next one down is the cryoarchive, and the donor requested this is only accessible to nonprofit entities. On the right-hand side are the different genetic mutations, both by gene and allele, and then mutation type, either transgene or knockout. And you can also indicate or register your interest in this gene. And then if you click on a strain, then this is the SDS, or the strain detail sheet, that appears. And this is where all the information is located in one place. So first you see the strain name, and then the stock number, and the citation ID. And the citation ID is the RRID. And it's very simple. You can merely click on this in order to capture it and then insert into a paper that you might be publishing. But then there's blocks of data here, as you see here on the gene information, where you can open up these individual windows to get gene details, a strain description, research applications, strain origin, also all the information you could possibly want in colony and husbandry information, and then finally the order request information. It includes the availability level of that strain, again, be it a live mouse, cryoarchivist germplasm, or ES cells, or so on, and then the fees. And I opened this one up because I know you'd be interested in fees. So this just happens to be one that is available as cryomaterial, but as a litter recovered from the cryoarchive at $2,829. Now, one might think, gosh, that's expensive. But if you think about it, if you tried to recreate this particular line in your laboratory, how many months of time and person effort hours it would be to actually recreate this. So it's actually a very nominal cost for a fully characterized, fully QC'd, and fully quality controlled line to ensure that it doesn't have any pathogens and it is the intended mutation. I mentioned also here on this slide is the conditions of use statement. You can also think of this as a digital shrink-wrapped MTA, but it's designed to provide only the absolute basics in terms of how your agreement and how you use this material. As you can see here, it's very straightforward. The material is not for use in human subjects. It's obtained from the MMRRC by the recipient and will not be further distributed to others or bred for fee-for-service purposes for a third party. That's obvious. And the recipient shall refer any requests for that line back to the MMRRC. And that type of information and those type of agreements, but it's as minimally invasive to your freedom as possible. And that's how we've tried to make it very, very simple for you. In terms of order fulfillment time, I put here a schedule for about how long it would take to receive a line after a request that it was cryo-recovered from the archive. So we see here, if we receive an order or request and conditions of use, or if it does require an MTA, then an MTA would need to be completed for some special collections. The order is in schedule, the order is placed in the center's order queue, and for international orders, we invoice and receive payment before we ship those. We start the resuscitation after 3 weeks, roughly, in which case we order the donors for IVF or transfer the cryopreserved embryos to recipients. Pups are then born, and by about 8 weeks, the pups can be weaned and genotyped, and the dam sent for a health screen. By 10 to 12 weeks, a health report is sent to the requesting investigator, and we request approval for shipment. And then by 12 to 16 weeks, the shipment is out the door and in your hands. So again, and also this is further facilitated if your institution has recognized the MMRRC as an approved vendor. So a very, very short timeline. And then finally, here, just to let you know all the ways you can contact us and follow us. There's customer service email as well as web support. We have phone number for North America, toll-free, and our international access phone number. And also, I encourage you to follow us on Twitter at MMRRC, and also our website, www.mmrrc.org. So thank you, and I'm happy to take any questions.
Thank you very much, Dr. Lloyd. I did receive one question in the private chat. This is from Dr. Bandrowski. With projects like KOMP represented here and many others, can you consider what are the places that you may be missing opportunities for MMRRC organisms or tissues? What I mean is that you are uniquely positioned in being able to create a set of knockout standard tissues for region validation, and this would be a real benefit to the antibody industry that is currently really struggling with validation and the reproducibility crisis, which some say is in large part of the antibody problem.
Yeah, I totally agree. So I think there's two ways to address that question, and both ways the MMRCs can help. Number one, in terms of antibody validation, those who have antibodies that need validation, of course, in tissues and living in mouse tissue is essential. Mouse lines that we already have in our archive can be browsed and searched for access to those that would be suitable for those validation studies. The MMRCs can, if the recipient doesn't require the live mice themselves, the MMRCs can process tissues from those mice and send those tissues to the recipient. In addition, the MMRCs also have, all of the MMRCs, the four, have the capability of conducting these type of studies in-house. That's an example of leveraging the host institution's expertise to assist investigators seeking antibody validation. So we're very, very flexible in however you want to do it. We can send the live mouse to the investigator that we have in our catalog, or the investigator can do so much as to even send the antibodies to an MMRC to do the validation in-house and everything in between. So that's one way. But there's a second way, and that is, remember, the four host institutions are also quite capable in genetic engineering and genome editing. So we can also help an investigator requiring antibody validation to actually generate the specific knockout that would be needed by the investigator to ensure validation of that antibody. So that mouse doesn't exist in the MMRC catalog, but the MMRC host institution can actually make it for the investigator. And again, then either send the mouse, send tissues, or conduct the antibody validation in-house. So I think the question is quite relevant. Antibodies do have a high hurdle for validation in terms of demonstrating that they are of use in their the specificity and sensitivity, and can't do that better than using a knockout mouse. And so the MMRC is available to help either with lines that are available, existing in the archive, or de novo mice that are made.
Excellent. I am not seeing any further questions in the group chat at this time. Dr. Lloyd, is there anything else you'd like to add?
Only that if anyone has any specific questions, please go ahead and use those email addresses to contact us, or call us, and we'll be happy to help you.
Excellent. Well, thank you very much, Dr. Lloyd. I'd just like to thank you for your time, and I'd like to thank all of our webinar participants who joined us on this holiday.